In this recording, we're going to discuss cellular transport. We've talked several times about the phospholipid bilayer of the plasma membrane. So now we're going to talk about how we move things in and out of the cell and go through the plasma membrane. As you recall, the phospholipid bilayer, or the cell membrane itself, the plasma membrane, has multiple components, but the biggest one are the phospholipids. We've talked about the phospholipids having both a hydrophilic and a hydrophobic component, the amphiphilic molecules. And the whole bilayer idea is to ensure that the hydrophilic heads point towards the watery environment and the hydrophobic tails are shielded from the watery environment. Well, the properties of the phospholipid bilayer also make it selectively permeable. And what does that mean? Okay, so permeable means we're able to move through the membrane. Okay, so we're permeable to the membrane. We can go through it. Selectively means we're kind of picky about what can just go right through the membrane. So not everything is going to be able to move between those phospholipids. If you cannot move straight through the phospholipids, we're going to have to do transport in a different way. Now, who decides and how do we decide who gets to move how? Okay. Well, it's going to be based on type of substance. So polar, nonpolar, ionic, large, small molecules, everybody moves just a little bit differently. Okay. Plasma membrane permeability to a substance. Okay. So how much do we want to move in and out? And then the concentration of the substance in the cytosol, or the intracellular fluid, compared to the extracellular fluid. Okay, so we'll talk about concentrations here in just a second. Now we have two main types of transport. We have passive and we have active. Anytime we're talking about passive transport, you are talking about moving solutes by not having to use ATP. Okay, so we do not have to use energy. Anytime you are talking about active transport, you are talking about moving solutes, and you are going to have to use ATP to move these. Now, the first type of passive transport we always mention is diffusion, just regular old diffusion. And we mention this because diffusion can happen both inside and outside your body, so it's easy to describe and it's easy to show you examples of. So diffusion is the movement of solutes, and these solutes are going to move from a high concentration to a low concentration. So I told you we'd be back to concentration, so here we are. So in our first image here on the left, okay, we have a beaker of water and it's got some glucose. We have a divider in the middle. You will notice that there is quite a bit of glucose on the left and just a few little tiny glucoses on the right. So we have a 50% glucose concentration on the left. So this is half water and half glucose. But over here on the right, we only have 5% glucose, okay, which would leave us over here with 95% water. Okay. Notice what happens when we remove our plastic divider. Quite a few of the molecules, the glucose molecules specifically on the left, hurry over to the right side, and they are going to continue to move until we have reached equilibrium. Now, equilibrium is reached when all of the molecules are fairly evenly spread out, and we refer to equilibrium as no net movement. Okay, so a net movement would be what we just did over here, a big left to right movement or a big right to left movement. Okay, but at equilibrium, we're not seeing that anymore because everybody's fairly evenly spread out. So this is how diffusion works. We're going to go from high concentration to low concentration until we hit equilibrium. Now, we can do this in our bodies. We can do simple or facilitated diffusion. For simple diffusion, we are going to move nonpolar solutes. These are usually very small solutes as well. Because they are nonpolar, they are able to, where's our picture? Here we go. We're going to take these little tiny solutes, these little nonpolars, and we're going to move them right through the membrane, right through the phospholipid. Okay, now notice that the orange molecules have a higher concentration outside, okay, in ECF versus inside. So we're going to move down our concentration gradient. We're going to go from high to low. So we're going to bring some more of these orange solutes inside. Okay. These little pink ones, however, we have a higher concentration inside compared to outside. So notice our arrows. We're going to move some of these pink solutes out to the extracellular fluid. Okay. So both of these molecules are going with their concentration gradient, or they're moving down their concentration gradient. You could hear either description. Okay. We're going to move from high to low concentrations. Notice we have not used any sort of protein helper. Notice that we have not used ATP, anything like that. We've just simply moved straight through the phospholipid bilayer. And the best example that I can give you for this is things like oxygen and carbon dioxide. So we would want to take oxygen that's outside of the cells, and we would want to move it inside. We would want to take carbon dioxide that's inside the cells and move it outside to get rid of it. Now, facilitated diffusion is just a slight bit different. Okay, so facilitated diffusion, facilitate. What does it mean to facilitate? It means to help or to assist. So now we're going to need a little bit of assistance to do our diffusion. Okay, so the types of molecules or solutes that we're going to move are now quite different. So we just moved nonpolars with simple diffusion. Now with facilitated diffusion, we're either moving polar solutes or charged solutes. Okay, so charged, think of like ions. Okay, and because these have different chemical properties compared to the plasma membrane, we are now going to need the help of a membrane protein. Okay, either a carrier or a channel protein. So you will notice that we could use channels. Okay, so here's our green and our orange channels. And notice that they're always open, but notice that they are specific. So our green channel is moving our green ions from high concentration to low concentration. Our orange channel is moving our orange ions from inside to outside, still down the concentration gradient, still going from a high concentration to low concentration. Notice that we are not moving any orange ones through our green channel. We are not moving any green ions through our orange channel. Okay. Now, same thing with carriers. Okay. The only difference is for carriers, our solute has to bind. Okay. So this little molecule came in, bound to the correct location, and now we've let that ion inside. Over here, our purple ion attached to the correct location of our carrier, and now we've opened up to the outside. We are still moving from a high to a low concentration. High to a low concentration. Okay, still moving down our concentration gradient. Okay, still have not used ATP, but okay, you can clearly see that these proteins, these carriers and these channels, we did not see those in simple diffusion. So that's our biggest difference. Okay, we need those proteins for facilitated diffusion. Now, osmosis. Okay, put a star by osmosis, y'all. All right, it's a difficult concept that we're going to have to master. Now, osmosis is water moving across the plasma membrane down its concentration gradient. So a lot of times you'll hear osmosis described as the diffusion of water, and that's fine. Okay, it's an adequate description. We are going to go from a high water to a low water concentration. Water passes through specific types of protein channels. They're called aquaporins. Okay, so aqua meaning water, porins being little pores. Okay. You do have just a little bit of water that will go right through the membrane itself, okay, as this picture shows, but most of the water moves through aquaporins. Okay. Now, here's why this gets so confusing. Any other time that we're talking about transport, simple diffusion, facilitated diffusion, any type of active transport, things like that, we're always talking about moving solutes or larger molecules. Okay. Well, osmosis, you're only talking about moving water. So what students will do, okay, is they'll get a little stuck on the solute and they'll say, okay, solutes, okay, so we have a bunch of solutes out here, we only have a little bit out here, okay, the water must be going outside the cell. Okay. And notice that's not the direction that the water's moving. Okay, because the solutes aren't moving, the water's moving. Okay, so if instead we looked at our little water molecule concentrations, you will notice that we have a few more outside than we do inside. So the water moves into the cell until we reach equilibrium. Okay, so when we talk about osmosis, we're talking about moving from high water to low water. Okay, which technically means you're moving from low solute to high solute. Okay, so if you get stuck in the bad habit of only ever looking at solute concentrations, osmosis could kind of confuse you a little bit. Now, why do we make a big deal out of this? Because, you know, your body's got a lot of water in it, and it moves around all the time. When we talk about osmosis, we're usually talking about tonicity, okay, which is a term used to compare your intracellular and your extracellular fluid concentrations. Now, in a normal everyday old human, your extracellular fluid and your intracellular fluid, they are isotonic to each other. Okay, iso meaning the same. Now, this means that you have no net water movement, which means you have no volume changes anywhere. And when we give these examples, we're always talking about little red blood cells. Okay, we're just always talking about little red blood cells. So, if your ECF, extracellular fluid, and your ICF, intracellular fluid, okay, if these are isotonic, so same concentration, then that means as many waters as you have on the left, you have the same amount on the right. As many solutes as you have on the left, you have the same amount on the right. So inside and outside are equal. Now notice this does not mean that water's not moving at all. Okay, so these little arrows are showing you that water is moving. Okay, but for each little molecule that goes inside the cell, we're going to have another little molecule go outside the cell. So everything is nice and equal. Okay, you do not have that net movement, that big movement in one direction or the other. That means this nice little red blood cell, okay, stays exactly the way it is. Now this is normal, okay, like this is what your body loves on an everyday basis. Okay, that isn't how it always is though. Okay, so sometimes things go wrong. If your blood cells are sitting in a hypertonic environment, okay, if your red blood cells are sitting in a hypertonic environment, then there is more solute concentration, okay, in your extracellular fluid than in your intracellular fluid. Okay, so we've got our picture here. Okay. So you will notice that we only have a few solutes inside. We have quite a few waters, but outside of our cell we have a lot of solutes, okay, compared to the waters. So what are we trying to do? We're trying to balance everything out. We're trying to basically dilute the solution. And instead of moving the solutes back inside, it's just easier to move the water. So you will notice that these little water molecules, you'll notice the direction of their arrows, okay? All of these solutes are going to pull that water out of the cell, okay? And notice what happens to this cell. It was nice and round and biconcave, and now it's all shriveled up, okay? So when you pull water out of the cell, it shrinks up, and we have a science term for that. It's called crenation. The cell crenates, okay? Your red blood cells don't work nearly as well with this shape. Okay. So hypertonic, okay, so like if you're really hyper, you get skinny because you run around a lot. Okay, so the cell shrinks because we've pulled out the water. Now hypotonic is the opposite. Okay, so now you have quite a few more solutes inside of your cell compared to outside of your cell. So a bunch of that water is going to move inside to dilute that solution. We're going to balance everything out. Okay. So notice our arrows are now moving in to our cell. When water gets pulled into the cell swells, okay? Just like you adding water to a water balloon. What happens if you add too much water to a water balloon? It pops. Well, your cells can pop as well. They will at the very least swell, but they could rupture, and we call that lyse or lysis, okay? It will not come as a shock to you to learn that your red blood cells do not work very well if they've all busted open. Okay? So both of these scenarios are not ideal for our bodies, and if they go on too long and your body cannot fix it, or if we do not get medical intervention, these can both be fatal. All right, so up until now, everything we've talked about, simple diffusion, facilitated diffusion, osmosis, it's all passive transport, okay? We did not use ATP ever. From here on out, we're talking about active transport, okay? We are moving things against their concentration gradients, okay? So remember, we had been moving things high to low, okay? Now we're gonna move things from low to high. We're going to force molecules where they don't necessarily want to go, and we're going to have to work at it, which means we're going to need some energy. We're going to have to use some ATP. So the best example that we have, and it's an example that comes up over and over and over, is the sodium-potassium pump. Okay, NAK, sodium-potassium pump. Your sodium-potassium pump is going to physically pump 3 sodiums outside of your cell. Every time it pushes 3 out, it's going to bring 2 potassiums back into the cell. And both of these molecules are going to be moving against their concentration gradient. Okay. So now we're going to go from a low concentration to a high concentration. And because these molecules are moving not necessarily where they want to, we have to work a little harder at it. And just like when you work a little harder, you have to use more energy. Well, so does this pump. And that energy is going to be good old ATP. Okay, so in our little image here, okay, so we are going to have our 3 sodiums attached. We are going to hydrolyze some ATP, get all that energy. Okay. Notice that when we do that, we have opened up the top of this protein carrier, and all of our 3 sodiums escape. All 3 of them. Okay. While we are open to the extracellular fluid, now we are also letting 2 potassiums bind. Okay. We have just a little bit of energy left to now open up to the inside, and those potassiums are going to be brought to the inside of our cell. Okay. Now, you are more than welcome to count all these sodiums and the potassiums on both sides. Okay. But you can quickly see that we've only got 2 sodiums inside, and we're trying to pump them all outside where most of the sodium already is. And we only had 2 or 3 potassiums outside, and we're still trying to bring those in where most of the potassiums are anyway. Again, keep in mind that this is the complete opposite of what we had been doing. We were moving from high to low, and now we are moving from low to high. It's a little bit harder to do that. We're having to physically pump ions, and that takes a little bit of work, a little bit of ATP. Now, last but not least, we can also move larger substances or larger quantities of substances. These are all active transport as well, okay, but these last few are going to require vesicles. Okay. So first of all, we have phagocytosis, which is cell eating. This happens when your cells ingest large particles from the extracellular fluid. And the best example we have is taking a white blood cell and it eating a bacteria. Okay, so notice that we have surrounded the bacteria in a vesicle. Okay, here's our nasty little bacteria in our vesicle. We are going to fuse with a lysosome, which does cellular digestion, and we are going to destroy that bacteria, which is kind of nice. Okay. Now, penocytosis is cell drinking. Okay. Instead of engulfing solid particles, now we are going to engulf fluid droplets. Okay, molecules that have been dissolved in the extracellular fluid. So we're going to surround all of these little particles. We've still formed a vesicle. Whatever these are, these could be nutrients or hormones, all kinds of good things. We can bring them inside and doing something with them. If we end up destroying these or making some sort of waste product, we can also get rid of any of the waste products as well. But phagocytosis, we're doing large solid particles. Penocytosis, we're doing little fluid droplets. So we eat solid things, we drink liquid things. Now, receptor mediated endocytosis. This one works very similarly to phagocytosis and penocytosis, but here's the difference. The difference is the receptor itself. Okay, so we have receptors on our cell surface that bind very particular molecules. Okay, when the particular molecule we're looking for binds to the particular receptor on the cell membrane, that is the signal that it's time to engulf everything in the area. Okay. Anytime you hear about any kind of transport, if it mentions receptors, it's receptor mediated. If it does not mention receptors, it's not receptor mediated endocytosis. Okay, so don't let this confuse you. This is how we move LDL particles into our cells. This is a type of bad transport. It's a type of bad cholesterol that we would like to get rid of. So we can bring it into the cell, kind of destroy it, and kind of get rid of the pieces. Now, not only can we bring things into the cell, but we can also get rid of things. And that's how exocytosis works. You are going to get rid of either large molecules or large quantities of molecules. And you may have heard this term secretion. Okay, it's the same idea. Okay, and exo would mean to exit the cell. Okay, endocytosis would be bringing something into the cell. So say we have made these little proteins inside of our cells, but they are supposed to function outside of the cell. We can fuse with the plasma membrane. We can open up to the outside, and the vesicle can release the contents into the extracellular fluid. Okay, and these will go on to do whatever their function is. It's just going to be outside of the cell. All right, just to recap, passive transport, we're not going to use ATP. We're going to go from a high to a low concentration until we've hit equilibrium. And we did our diffusion examples, both our facilitated and our simple diffusion, as well as osmosis. Active transport does require ATP because we're going from a low to a high concentration. We will not be reaching equilibrium here. And our examples are solute pumping, like the sodium-potassium pump, and then all of the endocytosis and exocytosis examples.